Mhm.
All good.
All right.
So back to what we were saying, we just wanted to have that icebreaker with you guys. We wanted you guys to name one thing you're excited about in medical school, and we wanted to congratulate you guys for making it this far and getting accepted into the faculty.
Yeah. Yeah, because we know that when we started, we were like really excited for everything and nothing at the same time. So if you could like text in the chat, or if you want to unmute your mic and introduce yourself and tell us what are you looking for forward most in medical school.
Tamara will be fielding the chat because while I'm sharing my screen, I wouldn't be able to see the chat or raised questions. So she'll just let me know if you guys have a question as we go throughout the session. Right. It's okay to not be excited about anything for medical school. I know it's a lot coming at you. It's fine.
Yeah. Maybe we should share our own, like what we were excited
okay,
about to like open the conversation.
I was excited for everything. You know, it was my dream to become a doctor since I was like 8. I know many of you probably feel the same. So I was excited to learn everything.
Yeah, same, but also when I used to watch a lot of medical shows, I was really interested in anatomy. So that was like something that I was really interested to start and see what that was about. So that was what I was excited for.
And Tamara put in this quote of the session. We'd like to start our sessions with a quote.
Yeah, just for motivational purposes, because medical school really can drain you. So the quote for the session is from Pirates of the Caribbean, one of the best movies, from Captain Jack Sparrow, and it's the problem is not the problem. The problem is your attitude about the problem. So in life, it's a guarantee that you will have problems. That is something you can't hide away from. So rather than focusing too much on the problem itself, maybe your perspective on it or how you're dealing with it would be better because that's something that's modifiable. So yeah, guys.
All right. And how the session is going to go, we're going to be taking you through molecular biology and cell signaling. We all have to start somewhere, right? And what embryology really is, it's the creation story. It's the story of us. So I'll be taking you through molecular biology, and Tamara will be taking you through the second half of the lecture where we focus on cell signaling. All right, so we're going to delve into DNA structure, but first here are a few things I want to make sure you guys know because I'm not certain of everyone's background knowledge, right? So let's have a nice conversation about the nucleus. We're starting really basic and simple because this is where your DNA is housed, right? So we're going to take a quick dive into the structure of the nucleus, components of the nucleus, and basic function. So you see this double membrane. Can you guys see my pointer or no? Or should I use a laser pointer? Should probably use a laser. Okay, here we go. Can you guys see this laser pointer? Tamara, you could probably indicate if they are shy to unmute their mic.
Yeah, I can see it. It's good.
Okay, thank you. All right, so you guys see this double membrane around it. This is the nucleus. This double membrane I'm speaking of. It's actually a phospholipid bilayer referred to as your nuclear envelope. Within the nuclear envelope, you have little proteins that are nuclear core complexes, right? It's shown as a gap here, but it's not necessarily a gap. It's a core complex that allows movement to and from the nucleus and into the cytoplasm, and that would be called your nuclear pore. So an example of something that would be going out via this nuclear pore would be like messenger RNA, mRNA, or something that would be going in would be like nucleotides. If you don't know what nucleotides are, don't worry, I'll be covering it in a bit. So there's two components to this double membrane. You have the outer membrane and your inner membrane. Now, this is what I want you to pay attention to, right? The outer membrane has ribosomes studded around it. And I want you to know this because RNA will come out of these nuclear pores onto the outer membrane, bind with a ribosome, and then get translated to the rough endoplasmic reticulum. And that's where translation and your protein synthesis would take place. Now, the next part of that envelope is your inner membrane, and there's a particular pathology associated with the inner membrane. It contains a very important protein that creates a structural framework for the nucleus. What pathology means, in case you don't know, it's clinical relevance, right? So when I say there's a particular pathology associated with it, there's something that can happen within a patient based on this, right? So it allows for interaction with chromatin where genes are expressed and replicated. And that protein that I was talking about is called lamins. Now, why is Ariane telling you all of this? Well, it's clinically significant, right? That there's a particular mutation in that lamin A, which is the protein on the inner membrane. And if that mutation is present, it would cause individuals to age very quickly. And that's called progeria. And I want you guys to note that down. Hopefully you have a pen and a notebook nearby. So inside of the nucleus now, there's a big component of a bunch of stuff, and that's called your nucleoplasm. It's labeled here. So there's two things in here, your nucleolus, you can see here, and this is where your ribosomal synthesis happens. So this is where your ribosomes are made. So remember this. It's also the ribosomes that can be shoved out of the nuclear pore that we just discussed. The next thing is this loopy kind of structure you see here, and it's called chromatin. And chromatin is made of histone proteins and DNA, right? Their combination works in a particular way. So there's positive and negative attraction to condense that DNA to fit all of that into the nucleus. Remember, your DNA would be so long. To fit all of that information inside of a nucleus, it has to be highly condensed, right? So to clarify on how that condensation works, it's the histones that are positively charged, and histones are just proteins, right, guys? And it's your DNA that would have a negatively charged phosphate backbone. We'll go into the structure of DNA to show you that sugar phosphate backbone, where it has the negative charge exactly. And that would generate an electrostatic attraction. Because guys, what happens when you have a positive and negative force? They just attract each other, right? So that's how it would condense DNA. I also included, you'll see along my slides, you'll have some highlighted terms. So nucleus holds all the genetic information. These are the terms I want you to be familiar with because you'll see them as you do your reading. And as we go through this journey of embryology, your creation story. So I want you guys to pay attention to genome. I said it has 23,000 genes in the human genome, and this is the entirety of the DNA's genetic sequence. And a gene is your segments or specific sequences within the genome that would code for a protein. All right, so we're going to move on to this squiggly thing. We want to zoom in here a little bit. Squiggly thing is called chromatin, right? Right, so I told you chromatin was histone DNA, heterochromatin, and euchromatin, right? Don't worry about heterochromatin and euchromatin, I'm going to explain now. Chromatin could be condensed into two forms. You have heterochromatin and euchromatin, right? I like to say heterochromatin, H, highly condensed. The H for here, highly condensed, right? So this is so condensed because of that electrostatic attraction I explained earlier. So it's really hard. If there's a histone complex and the DNA wraps around it, it's really hard for little enzymes to get in there and transcribe the DNA to make RNA. So there'll be no transcription in this type of chromatin. And I like to say E for expressing, right? And you'll see why I say expressing in a bit. So expressing for euchromatin, right? What I mean is that there's a weak attraction. So this heterochromatin has a strong attraction, so it holds it really tightly. Euchromatin, on the other hand, has a weak attraction between the histones and the DNA. So there's a space here where RNA polymerases could get in and make RNA. And this occurs because we want that part of the DNA to undergo transcription. So you get it? It's expressing because you could transcribe it and make RNA. So chromatin is important because whenever cells are undergoing replication, they want parent cells to easily be able to pass on DNA to daughter cells. So it condenses down into chromosomes. I want us to look at chromatin condensed down into chromosomes, right? But don't worry about the structure. I just want to let you know that this figure was taken from your Langman's textbook, chapter 1, right? I know you're seeing histone octama, nucleosomes, but we'll get to all of that. Don't worry. And you can feel free to ask me any questions as we go along if you don't understand something, okay? All right. So we're gonna take a closer look at this chromatin condensed down into chromosomes. So what happens is really, let me go back to slide 5. This chromatin, these squiggly loops, basically condenses down to make this chromosome, which is this X-like structure. I think you guys can see it on my screen here, right? So I want us to yank out some of that chromatin and look at it. All right. So you get this loopy kind of continuous fiber, and you keep getting smaller and smaller versions of it because we're examining it. So you get tighter coils, and do you see this red structure that DNA is wrapping around? What did I tell you DNA was made of? I told you DNA is made of your histone proteins, right? And there's a significance we need to talk about, and that's called your histone octama. Now, what the heck is an octama? Octama just means that there's 8 of something, right? Your octama has H2A, H2B, H3, and H4. Now, if we count, that's 4 things. 1, 2, 3, 4. But I told you it's an octama. Octama means 8, all right? So it means you need 2 of each. And that histone octama is what forms your histone complex that DNA is wrapping around. Now, when DNA wraps around that histone octama, that you can see close to number 3 here, when it wraps around twice, you call that a nucleosome. That's the particular word for it. So histones have a positive charge. I told you that because we were talking about that electrostatic attraction before. So when it has that opposing charge between DNA, which is negative, and that positive histone, that's how the chromatid would condense and wrap twice around, forming your nucleosome. That's just me recapping what I just said. Now, apart from this, when you get this nucleosome, which is a bead, apart from this histone, there's only one more I want you to remember, right? I know that I gave you a bunch for this histone octama, but there's one more called H1, histone 1, which is a linker protein. And when you get those nice beads of nucleosomes, what H1 does, it links those nucleosomes together. So you get kind of like I like to think of it like stringing together pearls. So H1 is your linker protein. So it strings it together and you get this kind of scaffolding product here. Now, why am I spending so much time explaining this to you? Well, nucleosomes, the histone proteins, and DNA could be modified via epigenetics to be silenced or enhanced. And this brings us to talking about epigenetics. The only reason I included this diagram is because I wanted you guys to get a full picture of what's happening with the steps of what's going on. Now, they don't necessarily test you on the fibers, but I do know in your reading sometimes you would come across 30 nanometer fiber to 50 nanometer fiber, and I want you guys to know where that happens. So that's the only reason I included this overview to how you get down to this chromosome, right? But you don't, like, the things that I'm focusing on, that's the stuff I want you to pay attention to because it's the stuff that you're going to be tested on. It's the high yield content we like to see. The rest of stuff is just background knowledge that I need you to know. All right, so let's talk about epigenetics now. I won't go through translation and transcription because I feel like you guys know it. However, you can reach out to me after the class and let me know, hey, Ariane, I want you to go through translation, transcription with me because I don't necessarily get it. I didn't get it when I did it in high school, right? And I would be fine with that, no problem. So let's talk about epigenetics. When DNA is uncoiled, right, you have zones that are exposed. Now, there's a particular zone. This figure 1.2, is also taken from your Langman's textbook, chapter 1, right? And Oh, well, this one's highlighted. So what did I say? We're paying close attention to those things that are highlighted, right? Now, there's a specific sequence when DNA is uncoiled, and it's called your Tata box. And what the TATA means, it's thymine, adenine, thymine, adenine. Those are nucleotides or nucleobases, right? And don't worry, right after this on like slide 10, we're going to get into nucleotides, nucleosides. Don't stress. All right, so this Tata box serves as a promoter, and this indicates your direction of transcription. Which of the two strands will be the template strand and has an initiator of transcription at this particular sequence, right? So what the polymerase looks for is this Tata box sequence at the promoter site, and it basically just tells it, hey guys, we can start transcription here. All right, and remember I told you when DNA is uncoiled, you have other zones. You have exons and introns. Those are some other zones you could see when it's uncoiled. So introns are your zones that are spliced out via alternative splicing by spliceosomes. Now, what is spliceosomes? I like to call spliceosomes fancy molecular scissors. So what they do is they just chop out the parts that you don't need, like snip, snip, and you get to keep some parts and exclude some parts, right? So I really like this diagram here, and it gives you a little overview of introns and exons. Now, introns are the parts that you're snipping, and they're not coding for anything, so you could basically leave them behind. It's your exons that are important, and you're going to take the exons and join them together because those are your coding regions, and those go on to making your proteins. Now, I really like this diagram here. So it shows you DNA and the copy of DNA, pre-mRNA that's uncoiled, to show you those zones that I told you about, the introns and exons. So you get your mRNA here, and you use your spliceosomes, your fancy molecular scissors, you go snip, snip, and you cut out your introns, you leave your introns behind, and now you join up your exons. And your exons is what goes on to give you proteins. Now, after pre-mRNA is formed, your spliceosomes come into play, and this is where spliceosomes are so important, your molecular scissors, right? Because they have the potential to create different proteins from the same gene, and that's known as splicing isoforms. Now, you guys did chemistry, right? So what are isoforms? Okay, we heard of isomers before, right? Where things have a different structural formula, but the same molecular formula, right? So it's kind of the same concept here, a splicing isoform. It's the same molecular formula, but it's a different structure. And that's how they would have the potential to create proteins from the same gene. And this is how your cells could make proteins from that same gene that's specific to that cell's function, because cells carry out a whole bunch of functions, right? Now, the only other important site that I want you to focus on that I will get to in slide 9 now, and we're going to speak about this poly-A addition sites. So we know that the Tata box says, hey guys, yeah, this is where you could start transcription. Well, the poly-A site says, okay, this is where you stop. Moving on here to slide 9, this is my poly-A tail, right? This slide is also, this figure right here, figure 1.3, is also taken from your Langman's textbook, right? So we're going to talk about this poly-A tail, and then we're going to go into enhancers and silencers. I'm just touching on these because I want you guys to know what it is. However, I've never seen it tested. It's just part of what you guys need to know. So I'm doing the high yield stuff, right? So what this poly-A tail is basically just a long sequence or a long chain of adenosine monophosphates, and adenine bases are just nucleotides. And why that's important is it makes this the RNA molecule more stable, and it prevents degradation. So it could go on to make a protein, because you don't want your exons when it's joined to just be like, okay, we're going to degrade now because the enzymes become too much. You need a stable product to form that end product, which is your proteins. So
Ariana? Hi.
yes.
Could you repeat what you said the poly A region is?
Oh yeah, no problem. So the poly A region is basically a long chain of adenine bases. It's adenosine
Yes.
monophosphates. So you
Mm-hmm.
guys know what a polymer is, right? Where it's
Yeah.
a long chain, right? So the monomers are those adenine bases. I'll go through what adenine is in a second because it's a nuclear base. And it basically just tells transcription, hey, you need to stop here when it encounters that sequence of this long sequence of adenine. There's usually stop codons close to the or embedded into that poly A tail region, and your stop codons could be, I want you guys to note down your stop codons. It's either UGA, you go away, you are gone, UAG, or you are away, UAA. And that's your specific sequences. And the U is for uracil, the A is for adenine, and the G is for guanine. We'll get to that in a second, and if you guys want, I'll re-mention it on the other slide where we focus on nucleotides. So your poly A tail is where RNA ends transcription, right? On mRNA, the poly A tail would protect the mRNA molecule from enzymatic degradation in the cytoplasm and aids in transcription termination, the export of mRNA from the nucleus, and translation. What I want you to focus on for this poly A tail is just knowing that, okay, it's composed of adenine monophosphates and that it says, hey, let's stop transcription here, okay? And you guys could note down that those stop codons if you want. Would you like me to repeat them?
Yes, please.
Okay. It's you go away, which is UGA, and you guys can feel free to stop me and tell me to repeat something, okay? Don't be shy. So it's you go away. That's the way I remember it because I need mnemonics, right? So it's you go away, you are gone, you are away. So you go away is UGA, you are away is UAA, and you are gone is UAG. Are we okay with that? Would you like me to repeat one more time, or do you guys got it?
I think we got it.
Okay, thank you for answering. All right, so we're going on to number 10. And this is the last thing that I'm going to overload you guys with is enhancers and silencers. Now, as I've mentioned, I have not seen enhancers and silencers on any tomato, you know, but it's something that you need to know. And how I like to think of enhancers and silencers are like parents. Your enhancers are your enabling parents. They will tell you, yeah, it's okay, you can go ahead, right? So what enhancers are, when you think of the word enhance is to make something better, right? So what enhancers are, it's a regulatory element on the DNA that would activate promoters. Now they don't, I thought it was important to tell you that, you know, when that DNA is uncoiled, they don't necessarily have to sit next to the promoter site. Remember that tartar box we just spoke about? They don't necessarily have to sit right next to the promoter, but they control the efficiency, the rate of transcription, and they alter that squiggly protein we were talking about, chromatin, to expose the promoter or to allow binding by transcription factors like RNA polymerase. There are a whole bunch of transcription factors, but the one important to this particular process is RNA polymerase, right? So separate enhancers can direct the same gene. So for example, they focused on your PAX6 transcription factor. So this is the transcription factor I would want you guys to know because it's in your Langman's textbook, chapter 1. So PAX6 transcription factor participates in pancreas eye neural tube development, right? And that would contain 3 separate enhancers. Basically, there's an enhancer for each of those sites because it would regulate the gene's expression in that appropriate tissue, right? So you have your enhancer for the pancreas, enhancer for the eye, enhancer for the neural tube. Now, I like this diagram again, and I included it because I wanted to show you that the enhancer is like an enabling parent, right? It says, all right, you want to go to the movies? Sure, we'll drop you there. It's fine. So basically, when the enhancers are here, they would allow transcription. So I don't want you guys to get confused. On this diagram, they say activators and repressors. Activator is another word for silencer. So activator is another word for enhancer, right? So when your activators are present and you don't have a silencer or a repressor, you can have transcription taking place. But if only one parent agrees, one enhancer, only one parent agrees and the other parent is like, well, they're on the fence or they're just not there at the time when you ask to go to the movies, you have little to no transcription taking place, right? Because you only have one enhancer or activator present, right? Now, do you guys get enhancers? Can I move on to silencers? I'm going to take silence as a yes.
Yes.
Hold on. All right. So just to clarify, just to clarify,
Go ahead.
the,
No
the
problem.
enhancer basically be a protein that allows or facilitates transcription. Example, RNA polymerase.
Yes, and your transcription factor here would be PAX6 in the example I gave you. That's an enhancer. Well, that's your transcription factor and you have an enhancer. But enhancer basically just proteins that are present. You have different proteins that need to be present for different things it's coding for, right? So, for
Protein makes the
example,
protein.
yeah. So it's just like, like when you have a plant and you need sunlight, water,
Mm-hmm.
and all those other additional elements for that plant to grow. Well, the enhancer is like those additional elements to allow transcription. Does that make sense to you?
Yeah, so the RNA polymerase is the enhancer and the transcription factor is the PAC6.
No. PAC6 is another transcription factor like RNA polymerase.
Oh, okay.
Yeah?
All right, thank you.
No problem. I just gave you two examples. So in transcription by itself, when it's not coding for anything, the genes are just transcribing, it would be RNA polymerase. And the example I gave you of another transcription factor is like PAC6, and that one would participate in your pancreas, eye, and neural tube. Okay?
Okay. All right.
Okay. Good. Let me know if you need me to explain anything else. So you have silencers now. And silencers work the exact same way as enhancers. They inhibit transcription, right? So silencers are your strict parents. Silencers are like, no, you cannot go to the movies and we're not allowing you to go there. So silencers allow a transcription factor to activate one gene while silencing another by binding to different enhancers. Thus, transcription factors themselves have a DNA binding domain that's specific to that region of DNA and a transactivating domain that would bind to a promoter or enhancer to activate or inhibit the genes regulated by these elements. There isn't a specific location, like I told you, where that silencer and enhancer has to sit. Now, Ariane, you said a lot of words and I don't get any of them. All right, well, I'm gonna explain, don't worry. So like I said, silencers are your strict parents. When you have enhancers binding here, once that silencer or repressor is present, you're not going to have any transcription taking place. And that's the basis of this that I want you to understand. Once a silencer is there, you don't have transcription taking place, right? And I also wanted you guys to understand that transcription factors themselves, like your RNA polymerase or your PAC6, those are transcription factors. They have like a little notch in them that could accept or bind to a region of DNA, and they have a trans-activating domain that binds to your promoter enhancer. So let's think of them as having two notches. One one notch binds to your DNA region, and the other one could bind to a transactivating domain, meaning it could bind to an enhancer that says, yeah, go ahead, or it can bind to a repressor or silencer that says, no, you can't. And that inhibits whether that gene is transcribed or not. Do we understand? And I promise this is the only heavy part of my lecture. We're going to go into the fun part now. So the fun part that I really wanted to talk to you guys about is your DNA structure, right? So DNA structure, we know that DNA is deoxyribonucleic acid, right? That's what it stands for. I just included that there in case you guys don't know. So DNA has your sugar phosphate backbone. And I really like this gray diagram right here. They have nitrogenous bases. Remember that A, G, U, all those things that I've been telling you. I have mnemonics for it, and you guys are going to get this in no time. So you have these nitrogenous bases here, which are nucleotides. And we know nucleotides could be strung together to make nucleic acids. So we're going to go through the constituents of DNA. And this is the easy stuff. So let's talk about pentose sugars. Now, pentose, pent means 5. You need a sugar, a phosphate, and a nitrogenous base to make a nucleotide monomer. And it's those nucleotide monomers that are strung together. This is a nucleotide monomer, by the way, guys. This sugar, phosphate, nucleic base, or right here, the A, or G, and this part that I'm kind of circling, if you guys can see it, that's your nucleotide monomer. And lots of nucleotide monomers are joined together to make your sugar phosphate backbone and DNA, right? We will go through why DNA has an anti-parallel arrangement when I talk to you guys about complementarity and base pairing, right? So let's talk about this pentose sugar. Pent means 5. And pentose sugars are really a ring sugar in the form of oxy, which is ribose, or deoxyribose. So there isn't a difference, there really isn't any difference between deoxyribose and ribose except for one atom. So in ribose, because it's oxygenated, it would have that OH group. And that OH group actually links to carbon number 3 right here. 1, 2, 3, right? So it would link to carbon number 3 here. So on carbon number 3 in riboses, you would have an OH group here. And in deoxyribose, we know it's not oxygenated, so you have an H right here. Right. And we also have a phosphate group. And this is what gives that DNA that negative charge to allow the histone interaction. And it's found on carbon number 5. 1, 2, 3, 4, 5, right? Or we can see it linked here on carbon number 5, right? And that's what, remember I said DNA is negatively charged when we were talking about electrostatic attractions before. So that's what conveys that negative charge to this sugar phosphate backbone, giving DNA that negative charge to interact with the histone proteins, right? So nucleosides make a nucleotide monomer. Let's talk about nucleosides and nucleotides, because I didn't know what the difference between a nucleoside and nucleotide was when I came in year 1, right? So there are 3 main differences. The first one that you can see right off the bat is that this one's missing a phosphate group and this one has the phosphate group on it, right? So nucleosides is your nitrogenous base added on to a pentose sugar, right? Your nitrogenous base, we'll go in onto what a nitrogenous base is on slide 14, right? Right after this. So your nucleoside is a subunit of nucleic acids. It phosphorylates to form a nucleotide. So basically, I like to think of nucleosides as the raw ingredient. And once it's phosphorylated and it's got that phosphate group added on, it now becomes a nucleotide. And nucleotides are the ones that make up that DNA backbone. So nucleotides are the ones that are joined on or are called building blocks of DNA, and they join at the 3' end. What do I mean by a 3' end? All right. So in DNA, you have ends, and the ends are pretty specific. They just show you direction. You have a 3' end and a 5' end. And that's to do with the DNA's anti-parallel arrangement, right? We're going to go into that in a little bit, but I just wanted to show you where the 3' end is and the 5' end. So this here would be their 3' end where it joins onto each other. Okay? So we said that your nucleoside has no phosphate group. It's just a base and your sugar, covalently linked, right? And nucleoside analogs are useful in antiviral or anti-cancer medications, but dysfunctional nucleotides can lead to cancer. So what I mean by that is your raw ingredient is like the nice guy. Your raw ingredients behaves nicely. You can use them to treat antivirals or as in anti-cancer drugs. You can use them to treat viruses or combat cancer in those types of medications. But when it's phosphorylated and it has the potential to be good or bad. So if it's good, it's going to go on to join them together and make DNA. But if it becomes dysfunctional, that could lead to cancer. And those are the 3 main differences. But these are not that important. I just wanted you guys to know the difference between a nucleoside and a nucleotide. As in, you're not tested on this a lot. So remember, I've been talking about nucleotides and nucleotides being strung up to interact and make DNA. Let me go back and let me just show you where the nucleotides are one more time to make sure that you're following me. These are your nucleotides. These nucleotide monomers here that I circled, right? And the nucleotides or nucleobases are these ATGC, right? So nucleotides make up nucleic acids, and we know that DNA is one of them. So now I promise you this is one of the easiest things you'll ever do in medical school, and you're going to get a question on Chargaff's rule, right? So pay attention here. So adenine loves to interact with thymine, and thymine loves to interact with adenine. Cytosine likes to interact with guanine. What do I mean by that? All right, let's start from scratch. What are nucleobases? Nucleobases come in 2 forms. They can either be purines or pyrimidines. Your purines have 2 rings. Your pyrimidines have 1 ring, right? Now, I need mnemonics, like I told you guys, to remember things, because in medical school, you have so much information being shoved at you that you really need something to help you remember. I liked this one. So this says pure silver, to remember your purines. Pure as in purines, get it? Purines, pure. And we know that the symbol for silver in the silver element in chemistry would be Ag. So pure silver, purines, adenine and guanine. So I put in a little diagram here that shows you your adenine, the structure of your adenine and your guanine. I should also note that you don't need to learn how to draw adenine and guanine, okay? I'm just showing you what they look like for you to appreciate that purines, to tell the difference, purines will have 2 rings and your pyrimidines have 1, right? My other mnemonic here is cut the pie. So I only included uracil here because it is a pyrimidine, but I want you guys to know that uracil is only in RNA. It is not used in DNA. Thymine is in DNA, okay? So pyrimidines, get it? Pyrimidines. And cut, cytosine, uracil, thymine. Clear on that? I think this one was simple. Okay? I'm assuming we are. So I told you before that adenine likes to interact with thymine, and cytosine and guanine like to interact with each other, right? And these are linked by hydrogen bonds. Chargaff, which is the scientist, came up with a rule. He said that there's a 1-to-1 stoichiometric ratio between purines and pyrimidine bases in both strands of DNA, and those bases should be complementary. He also said that if you have X amount of A, it should equal X amount of thymine. So if I had 1 A, I would have 1 thymine. So therefore, the amount of adenine is equivalent to the amount of thymine, and the amount of guanine I have will be equivalent to the amount of cytosine. Now, I know you guys are seeing the number of hydrogen bonds I've written here, but I'm going to show you, I'm going to go back to the gray diagram on slide number 12, right here, and it shows you that adenine, in fact, likes to pair with thymine, right? And we know that hydrogen bonds in general are weak bonds. Between A and T, or adenine and thymine, you have 2 hydrogen bonds. And between guanine to cytosine, you have 3 hydrogen bonds. So what does that mean? It means that the bond is easier to break between adenine and thymine than guanine and cytosine. Hydrogen bonds in general are weak bonds. And it's basically because they are electrostatic forces. But you guys know what is a strong bond? Between that phosphate and that sugar, right? So between that phosphate and the OH group, in the deoxyribose, there's a covalent bond that exists, and that's called your phosphodiester bond. You can take note of that bond, right? So this sugar phosphate backbone here is covalently linked by a phosphodiester bond, and between these base pairs, which is your nucleobases or nucleotides, they have hydrogen bonds holding them together. Now A to T is weakly held together between 2 hydrogen bonds, and G to C would have 3 hydrogen bonds. I also want you guys to take note of DNA, 12. Right, of DNA being an anti-parallel arrangement, right? So, okay, Ariane, stop it now. What do you mean anti-parallel? Well, when I was going through this with Tamara, she adeptly said that you could think of it as 2 snakes going in opposite directions, winding around each other to make this beautiful helical structure of DNA that we know and love and appreciate, right? So let me give you an example. This is adenine and this is thymine in the gray diagram we see here. We know that they have to pair up, but if they were sitting like in this 2D model together, right, they would just be right next to each other. You wouldn't have this helical wrapping going on. So what they really have to do is sit parallel or sideways to each other, like slant. Like you guys know that that slant where you know that you could put for the word or in your keyboard, that slant. That's kind of how adenine and thymine sit next to each other, and guanine and cytosine. So they have to run in 2 different directions. So your left strand of DNA actually runs in a 5 to 3 direction, and your right strand would run in a 3 to 5 direction. I know my arrow is wrong, I'll fix that, don't worry. So you have your 5 to 3 direction here and a 3 to 5 direction here. They need to be, you see how they flipped it so that the adenine could sit next to the thymine and the guanine could sit next to the cytosine, and it's kind of slanted, and that's because this is not a perfectly 2D structure. Your helical DNA is a 3D structure. I hope you guys could appreciate that. So we talked about complementarity and what this guy Shargaf said, right? And I told you that you would get a question on this. So let's go through a question. And this is actually one from a tomato people.
Excuse me.
So, yes?
Hello. Oh, could you please
Hi.
go back to the previous slide? I want to write something.
Okay, which one?
The one just before.
This one?
Yes.
Okay, no problem. You can let me know when you're finished.
Okay.
You guys shouldn't be shy to talk to me, you know. I don't bite, and I promise you that I'm really nice, and I'll go over it and try to explain until you get it. I don't care if it takes 10 times, I'll try. And I promise I won't get annoyed. So you could feel free to stop me at any point, okay?
Hey, I was just wondering if we're going to get the recording.
You are going to get the recording, yeah. It would be uploaded to a Google Drive, okay?
Okay, thanks. All
No
right.
problem.
I'm finished writing now, so you can change it.
You're
Thanks
finished
again.
it? Okay. Yes, no problem. So, all right. So we're going to go through a sample calculation for Shargaf's rule. And I want you guys to pay attention because I'm giving you one to do for yourself after this, okay? So get your calculator if you need it, or if you're good at mental maths. I wasn't good at mental maths to solve this, right? So I'm doing this one with you. When I read questions, I like to underline the important stuff right away. So a DNA molecule has 180 base pairs, right? Now that's important. They said base pairs. All right. So I know already that they're talking about a group like adenine and thymine together, or guanine and cytosine together. Now they said the DNA molecule has 180 base pairs and 20% adenine. How many cytosine nucleotides are present in this molecule of DNA? They mean, when they say nucleotides, they mean singly. By pairs, they mean they want them together, right? And nucleotides, they want them single. So they said a DNA molecule has 180 base pairs and 20% adenine. So let's write down the base pairs that are in DNA. You have adenine paired to thymine and guanine paired to cytosine. Now in the question, they told us you have 20% adenine. So what did that Shargaf man say again? He said if you had 20% adenine, you should have that amount of thymine. You guys remember from the slide we were just on here, right? Where he said adenine should pair to thymine, guanine to cytosine, purine to pyrimidine, 1-to-1 ratio. So the amount of adenine equals the amount of thymine, amount of guanine equals the amount of cytosine. I'm just repeating because I want you guys to get it. So they said 180 base pairs, 20% adenine, 20% thymine. So this is 40% if you add them up total. Now 100, which is 100% minus 40, is 60%. We're left with 60%, and we know that that 60% should be shared equally among our two remaining base pairs, guanine and cytosine. So 60 divided by 2 gives me 30. So you have 30% of each. Now they wanted to know how many cytosine nucleotides are present in this molecule. So they gave us 180 base pairs. So like I told you, base pairs, is it together? Adenine to thymine or guanine to cytosine, it's a pair. But they want nucleotides, singular. So we're going to multiply by 2 to get the nucleotides, right? So it's 1 because it's in a pair. So I'm separating it. So 180 multiplied by 2 gives me 360. So that's the total number of nucleotides in this DNA molecule that they're talking about, right? So they want the number of cytosine. We got 30%. They don't want percent, they want the amount. They want how many. So you take 360, which is your total number of nucleotides, and you multiply that by 0.3, and 0.3 is just the decimal figure of 30 over 100. So 360 by 0.3 gives you 108, right? So 108 is the number of cytosine nucleotides that are present in the molecule of DNA. All right, so I'm finished with this example. Does anyone have any questions? Do they want me to go through it again? Do we understand?
Could you do it one more time? You lost me at the 180 base pairs that you times by 2.
Okay, no problem. So you want me to go from the top or just from 180 base pairs? I'll go from the top if you want.
Well, just from the base pairs, but I don't know if anybody else wants you to go from the top.
Yeah, could you explain why you times it by 2?
Yes, no problem. All right. So they said base pairs, right? And when they say base pairs, they meant adenine pairs to thymine, guanine pairs to cytosine. So, all right, I want you guys to stick up 2 fingers, right? Like a peace sign, but they're together, like glued together. That's your 2 fingers, right? Together. Your index finger and your middle finger, right? You guys have them together? We following? Right. So that's your base pair. Your index finger or your pointer finger will be your adenine, and your middle finger will be your thymine. Right? Following me so far, right? So they said you have 180 base pairs. So that means you have 2 together, but they only want to find 1, which is a cytosine nucleotide. So basically, if you separate those fingers I asked you to stick up together, if you separate them and you form something like a peace sign, they're not too close together anymore. So you have your index finger is adenine by itself, and then you have your middle finger that's thymine by itself. So remember, it's 2 things that make up a pair. Now it's by itself. That adenine alone is 1 nucleotide. So that's why I multiplied it by 2. Because they said you have 180 base pairs. It doesn't matter if it's adenine and thymine or guanine and cytosine. They said it's the number of pairs in total. You could be either adenine and thymine or guanine and cytosine. Do you understand why I multiplied by 2?
Yes, thank you.
Okay, cool. So you take 180 and you multiply by 2 and you get 360. I don't want you guys to just tell me, okay, you get it. If you really don't, I'll try again to explain it in a different way. So it's 180 multiplied by 2 and you get 360 because they want the number of nucleotides by itself. So remember, I asked you to stick up your fingers. So they want like your fingers by itself. They don't want them joined together. You get what I'm trying to say? So they basically take 180 multiplied by 2 to get that number of singular nucleotides, that one finger, singular nucleotides. So you get 360. And then you take that 360, they ask for cytosine. Now we had a percentage. So for example, if I asked you, we already know the number for cytosine, right? So if I had asked you from this one question, I had asked you, hey guys, you have the same numbers, but I want to know how many nucleotides are present in the molecule of DNA. How would you work that out? Can someone unmute their mic? I want to know how much adenine nucleotides are in the molecule. How would you work that out?
I would find 20% of 360.
Right, exactly. So you take the 180, you multiply by 2 to find the total number of nucleotides, and we already know the quantity. So you just find 20% of 360. And that's your answer. Do we get it?
Yes, yes.
Yes, we follow? Okay. Please don't tell me yes and you really don't, okay? I want you guys to get it because you're going to get one on your exam. So you're not harming me, you're going to be harming yourself if you really don't know how to do it.
Can I
Get it?
can I get an opportunity to like explain it and see if I understand it properly?
Yeah, sure, go ahead.
All right, so we want to find the number of cytosine bases, right? Bases cytosine out of the 4 in total that we have. So they told us that we have 180 base pairs, which means that
Okay.
there's 360 bases in total present. since we're peers now.
Exactly.
All right.
You've been good so far.
And based on, I don't know if I'm going to pronounce this right, Shagarov's rule,
Yeah,
there's a
Sharkas rule.
Shagarov's or
Don't worry,
Shagarov's.
pronunciation is fine. It's fine.
Okay, all right. So based on Shagarov's rule, it's a 1-to-1 mole ratio between each bonded, or I guess you wouldn't call it bonded, each interacting basis. So
Right. Yeah,
one
they're
ad
paired, right?
okay. So wait, one adenosine would make a hydrogen bond with one thymine. So if we know
Great.
that,
Well, it's two hydrogen bonds between it, right?
yeah,
But
I don't get that
all right,
part as
good.
yet. I don't memorize that as yet. All right. So that
That's
means
okay.
for the 20% of adenine that we have, that would have a equal 20% of thymine. So in
Good job.
total, okay. So in total we have 40% of the A and T bases, which
Right.
leaves the remaining amount of bases to be 60%, that being between the
Yes.
cytosine and the guanine present.
Right. And we know that they're equal, right? They're shared equally. So that's how you divide by 2 to get the purity.
Because of Shagarov's rule. All right.
Yes.
So since it's equal, that means it's 30% of each of them, and therefore we just need to find 30% of the total amount, 360. And how much
Yes.
is that?
108.
108. Okay, thank you.
Yeah, no problem. Good job. Thank you for being brave and unmuting to explain. So everyone gets it?
Both sides same is 108. All right. Maths.
Does everyone else get it?
It makes sense.
Yes.
It makes sense?
Understand. Yes, understand.
Yes? Okay, well, I'm really going to see.
Yeah, I get it.
Wait, what?
Low-key relying
Did
on the
someone say they
chemistry.
don't understand? Did someone say they don't understand?
No, I think everybody was saying that they got it.
Okay, well, I'm actually going to see if you guys get it now, because I'm giving you one to do on your own. So I'm going to read it with you, and I'm not going to do it with you, though. I'm just going to read it for you, and you're going to tell me the answer. You don't need to show the answer. You can just put it in the chat, and Tamara will let me know. Right? So the GC content, they mean guanine, cytosine. Now they paired them, right? Of a DNA molecule is 60%. What are the percentages of the 4 bases? This is actually a question from a tomato, a past paper, right? I don't want you guys going out there saying that you have past papers to any of your lecturers, please, because then that'll make things hard for everyone else.
Can I answer?
Huh?
Can I answer the question?
Yes, you can answer it.
Is it B?
Does everyone else think it's B?
Yes.
Yes.
Because they
Yes,
said
I think it's
60% is GC, so you have to split that 60% into 30 and 30. And then the remainder of the 60 is 40, which splits into 20 and 20. So
Yeah, good job. It is B. Thank you so much for
Yeah, go
explaining as well.
Chelsea. That was really good.
That was so good. Thank you. All right, so you guys got what Chelsea said, right? But just in case, I'm just gonna say it one more time. So you have 60%. This shot graph, guys, say it's in a 1-to-1 ratio. So you split that 60 in 2, so you have 30% and 30%. Now the percentages that remain is 100 minus 60, which is 40. We have an adenine and thymine to account for. 40 divided by 2, 20% and 20%. Now why E wasn't the answer, guys?
because E had U in it, and U isn't present in a DNA molecule.
Yes, that's so good. Thank you. I feel so happy that you guys are actually understanding and you know that uracil would be only for RNA, right? All right. And that is the end of my section on molecular biology. Is there any questions, or would you guys like a break time if you need one or you would like one before Tamara takes you through the wonderful world of cell signaling? You guys have any questions?
Jimmy and Crystal.
Okay, go ahead.
Sorry about that. Never mind. Sorry about that.
Oh, okay.
Okay.
I want, I want,
Just a quick question. You
Go
keep
ahead.
mentioning the chat, but am I the only one not seeing it? Because I don't have the option.
I'm not
Um,
seeing the chat either.
do you see like
Oh. Tamara, could you guide them through it? Basically, when you open, you should see a little bubble that has like 3 lines in it, looking like a chat button. Do you guys see that on the top right hand of your screen?
No.
No?
hold
Right.
up.
Let me send you a message and let me see if you guys get it. Right? Are you guys seeing this? Are you guys seeing my screen currently?
Yeah, but so we don't have, well, I don't have that option. I have the participant option and the option to raise my hand, but I don't have the one in the middle for the chat.
Right, same. I see that you're saying that it's only available for team members. And I guess we had a guest link, maybe that's why.
I didn't join with a guest link.
Great. We'll try to sort that out for next class, okay, guys?
Okay.
But if you want, you don't need to be shy. I feel like I'm approachable. You could unmute your mics and talk to me. I would love talking to you guys.
Hi.
Okay. Hi.
Can you hear me?
I can hear
I
you.
just have
Go
a
ahead.
question about the enhancers and silencers.
Yes.
If there's a silencer present, transcription wouldn't take place at all. Like, does it prevent the transcription of a particular gene, or will transcription just not occur at all?
Oh, well, transcription will still occur, but it would be transcription of a particular gene that would not occur at all.
Okay, perfect. Thank
And
you.
that's really useful, the enhancers and silencers. I'm glad that you brought that up. It's really useful for enhancers and silencers. So, for example, let's say in your brain you have a chemical imbalance, right? And it has a particular gene and it says, okay, you need to stop making that gene. That's where a silencer would come in really handy to say, well, we have too much of that and you're going to give me an imbalance. So you need to stop it. Do you understand?
Yes, I understand.
Okay, great. All right. Does anyone else have any
Giselle,
questions?
are you okay? I see Giselle's raising her hand.
Okay,
Yes.
Giselle, go.
My question is, you had mentioned a type of bond in the
Yes.
connection to DNA, and you said it was important to note. Can you repeat what that kind of bond is?
Yes. I'm going to go back on this slide just so you understand what I'm talking about, okay? We're going back 1 minute. Just give me 1 second to scroll through back these slides. Right, it should be on slide 12. Slide 12 you're talking about, right? Okay, good. So here we have your sugar phosphate backbone. I wanted you guys to note that there is a covalent bond between the sugar and phosphate, and that's called a phosphodiester bond. Right? And I also wanted you guys to note that between adenine and thymine, those nuclear bases, that you have hydrogen bonds. So in the sugar phosphate backbone is where you would have the phosphodiester bond or covalent bond. It's a covalent bond, that's just the name of the bond, right? Here, and in the base pairs, you would have your hydrogen bonds. All clear? So if I were to show you on this DNA helical structure, this part, you see this dark gray part that I'm kind of going around with my laser pointer here? Right. This one is basically where you have that phosphodiester bond or those covalent bonds happening. And the ones here in the middle, like little bridges joining, where you see that join here, 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 those are hydrogen bonds. All good?
Also, Kira, are you okay?
Are you okay?
I see Kira Christopher raising her hand.
Hi, yeah, I had a question. Can you just go through the difference between the enhancers and the transcripting factor again, please?
Okay. All right, well, enhancers and silencers, I didn't want to scare you guys. They're not that important in terms of you're not going to get tested on it. At least I don't think so. I've never seen it before, but just for your personal understanding, I am going to go through it. Don't worry. All right. So do you want me to go through enhancers and silencers or just enhancers? You let me know.
Just the difference between a transcripting factor and enhancer.
Okay. So an enhancer is basically, so your transcription factor is what comes in and does the transcription of DNA itself. And your enhancer is like that element, that complementary element that needs to accompany the transcription factor to say, yes, you can go ahead and transcribe, or no, you can't transcribe. Does that make it simpler for you? Where you have...
So the enhancer doesn't have to be close to it, right? It could be way up along the DNA.
Yeah, they don't have to necessarily sit next to the promoter. Because remember, I was talking about, let me go through that. So we were talking about this region here called the tartar box, and that's a promoter region, right? So your promoter region is that is the region that says this sequence thymine, adenine, thymine, adenine. So your transcription factor will scan through that whole copy of DNA looking for this starter box sequence. And when it finds it, it's going to be like, yes, here, guys, this is where we need to start transcribing, right? So this is where transcription starts. And for your enhancer, it doesn't necessarily need to sit in that tartar box region, right? It could be anywhere along the strand after it or before it, right? Once it's there. Like, for example, like I gave that example of a plant, which I came up with totally on the spot, but you have a plant that needs these other elements to come in, like your sunlight, water, for it to grow, right? So the same thing. Your enhancer are like those regulatory elements, like your sunlight and water and whatever. Obviously not the same for this because it's DNA, it's not a plant, but it's those regulatory elements that you need in addition to that transcription factor to say, yes, you can transcribe. And I like to think of them as the word enhance. Enhance is to allow, right? Or enable, right? And I like to think of them as enabling parents. They're not strict. They say, yes, we'll allow you to go to the movies today, right? And your silencers are your strict parents. They say, no, you can't do that. Do you understand?
Yes, I do. Thank you so much.
You do? All right, no problem. I'm happy.
Also, Jeremiah, you have up your hand.
Jeremiah.
Yes, I wanted to clarify on the charge of the DNA. You said that it's negative, but I wanted to just go over why exactly gain the negative charge. I see that the base is negative, but I'm not exactly sure why.
Okay, no problem. So the reason DNA is negative is because of the phosphate that is on the DNA, right? Now, the reason I said that the phosphate is negative is because the phosphate head itself that joins onto the sugar phosphate backbone, it kind of encases the whole molecule, right? So if I have, for example, my phone, right? And I have a phone case on my phone, and the phone case covers the screen and the back. And that phone case has a negative charge all over it. Like, let's say the phone case is made out of metal, right? And I have a magnet above the phone case. The phone case would be attracted to the metal, even though what's inside it may have opposing charges. Do you understand what I mean? Because it's the thing that's encasing it.
Yeah, so essentially, if it has a charge, then everything else adapts that charge.
No. What I'm trying to say is the sugar phosphate backbone, this gray structure here, this one that loops around, that has a negative charge because of the phosphate group that's on it. Because nucleotides themselves have a phosphate group. Remember, I told you the difference between nucleosides and nucleotides. It's because of that nucleotide, which is this whole monomer, basically all the monomer subunits, which is this part here, has a negative charge because of that phosphate that's on it, right? And that phosphate is what gives DNA the negative charge to allow that histone interaction. And histones themselves are positive, right? And you know what would happen between opposing forces, right? Positive and negative would attract. and it's found on the carbon number 5, right?
Yeah. So going from hue-chromatin to chromatin, heterochromatin rather, why is it that the intensity of that negative charge or the attraction decreases?
All right. So let me go over that one more time. Let me go back to heterochromatin and euchromatin, right? So that you understand. But you do understand why DNA has a negative charge, right? Because of the phosphate group?
Yeah.
Cool. Let's go through chromatin. Let me get to that section. Sorry. It's taking me a while to scroll. All right. All right. So I told you that I like to think of, so chromatin can exist in 2 forms, right? Heterochromatin or euchromatin. Now, heterochromatin is, I like to think of the H for highly condensed, right? Now, what that means is that it's so condensed because of that electrostatic attraction between the histone and the DNA. So RNA could get in there to transcribe DNA to make RNA, the polymerase, I mean. So there'll be no transcription. All right, let me re-explain that. You have heterochromatin, H, highly condensed. You get that so far? H for highly condensed.
Yeah.
So if something's highly condensed, it wraps so tight around each other. You could put your hands together and like clasp them together and squish them tight. You think that an RNA polymerase could get in there when you're squishing them so tight together that they just basically stuck on each other? Your RNA polymerase can't get in there. So you have no transcription happening there. And for your euchromatin is a weak attraction because in that form of chromatin, it has a weaker attraction between the histone and the DNA, right? Because of that weak attraction, okay, remember I told you to grip your hands together, loosen them a little bit. That would be your euchromatin, and I like to say this one is expressing, E for expressing. So if it's looser, right, there's a space, and that space is where RNA polymerase could get to come in, and basically we want that to happen, right? Because we would want RNA polymerase to get in there and make RNAs because we want that part of DNA to undergo transcription. So that's why I called it expressing, because you can transcribe here. Do you understand?
Yes, thank you.
Yes. Okay, no problem. Anyone else have a question that they would like me to answer?
Yeah, I have a question. Well, multiple questions actually, but the first one is on the structure of the chromatin, basically. So
okay, we're going back to chromatin. All right.
The chromatin you said is made up of
DNA.
genes, right?
Right.
DNA. Okay, genes are made up of chromatin.
Yes.
Right? And chromatin is made up of histones and
DNA. Right.
Right. So DNA is the smallest. Well, nucleotides would be the smallest structures, basically.
I would like to say that your chromatin is the smallest structure.
Okay, so it's DNA, then
let me go into this diagram. Wait, let me go into this diagram. Okay, so let me try to show you. So you have your chromatin, that's the loopy one that we saw in the nucleus, right? And that chromatin goes on to make a chromosome, and your chromosome is this X structure here. Do you get that?
Right.
Right. And in the process of making that X structure, this is chromatin here, right? And in the process of making that, chromatin is the smallest, right? And then in the process of making that, when it wraps around that histone octamer we spoke about earlier, it would create a nucleosome, and then the nucleosome highly condenses and condenses and condenses because of that electrostatic attraction. That's what's going on there, that electrostatic attraction we spoke about between the histones and the DNA. Histones being positively charged, DNA being negatively charged, and they fold up and produce these fibers, and they get so tightly coiled around each other that they form this chromosome. And this chromosome is really important in cell replication because that's how parent cells would pass on to their daughter cells.
Okay.
You sure you understand?
Yeah.
All right.
The
I'm
second
happy you do.
question
Go for it.
was about the charges. You said that DNA was negatively charged and the histones were positively charged.
Yes.
So is it that the sugar, the pentose sugar and the nucleotide base, they have neutral charges?
Right. So your nucleotide bases, they kind of, what happens is they do have charges on them, right? But they kind of cancel each other out because the bigger, all right. You all did forces before, right? In like physics.
Yeah.
If you have a bigger opposing force and you have a smaller one, for example, I have a box and it has to be towed by boat. And I have a big speedboat and I have a little dinghy on one side. And they both attached to the box. Right? And they're pulling in opposite directions. Which one you think would pull the box in that direction?
The bigger box.
Speedboat, right? Right?
Yeah.
So they do have charges on them, but the charge is really not important at all. Because of the bigger opposing charges. It's because that sugar phosphate backbone is encasing those bases. Most of the bases are actually negative themselves, like the adenine, thymine, most of them are actually negative themselves. I'm trying to go back to that slide. Give me 1 second so that you could fully understand. Yeah, okay, this one, 12. Oh, why did it go so far? No. Right, guys. 12. Okay. To give you a visual picture here with slide number 12. All right, so your adenine, thymine, guanine, most nuclear bases are negatively charged, especially lysine, arginine, all those. But the ones that are important for DNA, these are negatively charged. But the thing that's encasing them that I'm trying to tell you is that you see that these are just little bridges within it. But what's surrounding it is that helical structure, that sugar phosphate backbone. So it's that phosphate that really conveys that big negative charge. So you could think of the phosphate as a speedboat, and the base pairs, they do have a negative charge, so they are pulling, but they are the dinghies. They're not winning. It's really that phosphate that's carrying that charge. Do you understand?
Okay, so because the phosphate is the one that's bonding, that's the only charge that matters, basically.
Yes.
Okay.
But I wouldn't say it's because it's bonding. It's really because it's outside. I really wanted you guys to get that concept, like, for example, I don't know, like you have your phone, right? And let's say your phone, it doesn't matter if it's positively or negatively charged, right? And then you have a metal case encasing your whole phone, right? So your metal case, let's say that has a negative charge and the magnet has the positive charge, it would attract, right? But it doesn't matter what the phone itself is, but it's the case that's necessarily being attracted.
Okay.
I'm sorry. So you're saying that what is at the end determines what binds to the complex, so to speak.
I
Or the
wouldn't say what's at
charge?
the end, but I would say the backbone, yeah. The backbone.
Okay, okay, all right, thank you.
Yeah.
Okay, so my last question is about the enhancers and silencers and transcription factors.
Right. Going on to
So
enhancers and silencers. Go on.
yeah, so is it that you said that on the chromatin, the transcription factor looks for the promoter region, and you said that the enhancers,
When the DNA is uncoiled, right? You have a transcription factor like RNA polymerase. They have one here, RNA polymerase 2 mainly, right? That looks for this starter box region to bind it, to find this transcription initiation site.
right? So is it that the enhancers and silencers are on the transcription factor, or they are on the chromatin, but just not close to the promoter
They are
region?
on this. No, they are on the strand of DNA.
So on the chromatin.
Yes.
Okay. Okay, that's it.
No problem. Does anyone have any other questions, or are we clear? Are we good?
Clear so far.
Clear? Okay, I'm happy you guys are clear. But I don't want you all to focus too much on the details of enhancers and silencers. Like I said, I've literally never seen a question with it before. This will be on your paper. Well, I wouldn't say will be because I don't set your paper, but we did have this in our year, a question like this. This was actually the question, I believe. I'm gonna allow Tamara to take over now. Do you guys want a break? Or we're clear to move on to cell signaling? Guys?
Can
All
move
right, we're
on.
gonna take some minutes as we
Yeah,
can move, right?
Sorry?
Good. All right, so I'm gonna pass you over to Tamara, who'll take you through cell signaling.
All right, so hey guys, just reiterating my name. I'm Tamara Mercurius, and we're doing cell signaling today. Just two things to note about cell signaling is that really and truly it's not an important topic in the sense of high yield, but I will say that it is a foundation to understand a lot of concepts in embryology, but not really. So don't stress it. It's really de-stressor right now, and hopefully can go through this quickly. Also, note the tomatoes do not ever have these questions. There is no questions in existence that I've ever seen that is pertaining to cell signaling. Okay, so we can like relax. I can't turn the slide, Ariane.
All right, hold on, I'll switch them for you.
Thank you. All right, so we're going to start with what is cell signaling. Need to understand that before we move forward into other stuff. So basically, cell signaling is a form of communicating between cells that involves secretion of signal molecules that are accepted by specific cell receptors. So what is all of this saying? Basically, it's cells communicating. So just like how you would talk to your friend or your mom, cells talk to each other. That's how they know what they need to do. So in my little nice cute diagram right here that I did not draw, there is a sending cell. So say that this cell wants to send a message to the target cell. So let's say that the message is want to ask the target cell to go to the corner shop to buy alpha bread. So that's what the sending cell wants to send to the target cell, right? So it's gonna secrete these little circles that you see right here. They're known as ligands, aka signaling molecules. So that the message was corner shop, half a bread. So now it's going to the target cell, right? And this little thing here that's bluish, that's the receptor. And it's picking up the ligands and then internalizing it. And then that will be a response produced. So nothing hard, it's just cells communicating to each other. And that's how they do it. All right, so now we know what cell signaling is. Why is this even important? Why are we learning this? To put it shortly, I took this straight from the Langmans. Love that book. This is not my words, but it is essential for induction, and induction is basically how cells would direct other cells into developing. And you're gonna see that a lot in embryology, where other cells is gonna tell other cells, oh, build a nose, build a arm. Yeah, so that's induction. And it's also used for the conference of competency to respond. What is all of that mumbo jumbo saying? Basically, the target cell itself has a ability, a certain magnitude to respond to the signals that are being given out to it. So these ligand signals right here, like how well can this target cell like perform the task that was asked of it, basically. And also, lastly, it's essential for the crosstalk between the inducing cell, which could be seen as the sending cell, and the responding cell, which could be seen as the target cell. And here again, a little cute diagram, nice and sweet, with like the ligand binding to the receptor. So all of this would have been the target cell, right? So the ligand is binding to the receptor, and then you see a lot of stuff going on inside, and then boom, a response. Um, Everybody gets that? So we know what cell signaling is and the importance of it. We're good? Okay. I'll take you guys silence as we're good. All right. So you can switch to the next page. All right. So before getting into the meat of the matter, just defining some outer line terminologies that I've been using. So I use the terminology ligand very loosely. And basically, what a ligand is simply is the signaling molecule. And I love when things in medicine is actually simple because it's very hard to find something simple in medicine. So ligand is a signaling, and it can be hydrophobic or hydrophilic. So if it's hydrophobic, just to break it down, hydro means water, and phobic would be like a fear of something. So basically, it not really like the water thing. It doesn't agree with water, or it can be hydrophilic. Philic itself, that Latin word means a fondness towards something or loving. So that would be water loving. So you get it, hydrophobic, hydrophilic. Oh, somebody. Okay. So with the understanding now, when you get to the characteristics of the two, so a hydrophobic ligand characteristic. Okay, so it needs a hydrophilic carrier protein to carry the cell of interest. Oh, sorry guys, my Wi-Fi almost tripped out a while ago. Sorry, but yeah, what I'm trying to say is that it needs a hydrophilic carrier protein to carry the cell of interest, and basically what's happening here is that the ligand that I said that was going to be secreted from the sending cell, it's gonna secrete it into the extracellular matrix. And I don't know if you guys remember, but in the extracellular matrix, there is like tons of water. So can you imagine if you don't like water and you're in tons of it? It must be so hard for you to transfer your little message to the target cell. So because of that, our nice bodies created a hydrophilic carrier that is able to transport the ligand to the target cell. All right. So next point now about hydrophobic ligands is that it can diffuse past plasma membranes. Let me see if I can draw something. Can I? Oh. Aaron, do you want to draw it for me? I just want to draw the plasma membrane with the hydrophobic head and the hydrophilic head and the hydrophobic tail.
Yeah, no problem. I can try to draw it for you. Let me try to find a pen.
All right. So while Aaron drawing that, what I wanted to say is just to remind you guys of the fluid mosaic model of the plasma membrane, and just a reminder that the head was hydrophilic and it was in contact with the extracellular matrix and the cytoplasm, and then the tail was hydrophobic. So because the tail was hydrophobic, the hydrophobic ligand was able to pass through the plasma membrane. You guys understand that? So it's hydrophobic, them greed, them having a good time, so they can easily pass through the plasma membrane. Arian, that's enough. That's enough. That's enough. That's good. Thank you. So yeah, they can just easily pass through the plasma membrane because of the nice hydrophobic tails. All right, keeping it simple. All right, so the next thing now is that they bind to the receptors inside the cell. So the hydrophobic ligands, that's where they typically bind. Reason being is that most of the receptors that are inside of the cell, which are intracellular receptors, are small and hydrophobic themselves. So really, hydrophobic ligands can pass through the plasma membrane and be able to reach to these receptors. So that's the long and short of it. So now that we know how the hydrophobic ligand characteristics are, it's easy to like deduce what the hydrophilic would have looked like. So first of all, it's going to arrive to the cell of interest themselves. So they can do it independently. They don't need help. Because if they're in the extracellular matrix full of water, I think 60%, they're gonna have a grand time. This is nothing to them. So they'll reach the target cell on their own. Also, they cannot diffuse past the plasma membrane because as I just said, like the hydrophobic tail and hydrophilic ligand, that's not good. So they're not able to pass through the plasma membrane. But again, our bodies are constantly like doing the most for us, and we'll have to applaud it. The hydrophilic ligand is able to bind to the transmembrane receptor itself. So you can see on this little cute diagram right here, there's like this purple receptor here, and then that blue thing that's binding to it, that would be the ligand, and it's on the cell surface. Why is it called transmembrane is because there's a cell surface part, there's part that goes through the plasma membrane, and there's also a part that's in the cytoplasm. So that's what the hydrophilic ligands would have binded to. Are we good with this? Do I need to explain anything? Are we good?
Can you
Good.
explain the binding to the transmembrane receptors again, please?
Oh, all right. So for hydrophilic ligands, what I'm trying to say is that remember that they can't go past the plasma membrane because of the hydrophobic tail. So what there is a transmembrane receptor, and it has like a three-part domain. So there's a cell surface. That's where the ligand would have binded to. That's the receptor that you're seeing. And then there's parts of that same receptor that goes through the plasma membrane. And then at the end, on the other end, would be it going through the cytoplasm. So it's like a long thing, just from the cell surface through the plasma membrane and then into the cytoplasm. You understand that?
Yes, thank you.
Okay, no problem. All right. So the next terminology that we need to define is a receptor. This is quick and sweet. So it's just ligands bind to this, and as you guys know, the ligand is the signaling molecule. So honestly, receptor, the ligand binds to it. There's different examples of receptors, and I just gave you three, but there's tons more. But these are the major ones that are gonna pop up in like MoleMed and Cell Bio for you. And that's G coupled receptors, enzyme coupled receptors, and ion channel receptors. I didn't go into them because it's not necessarily a major deal in embryology, but I just want you guys to know of it. So yeah, nothing hard. All right. So we're going to the types of cell signaling, and we finally reached the meat of the matter. It was a quick journey, so we can everything that we just learned a while ago, we can just put them into place and apply the knowledge. So first one is autocrine signaling, and it's basically cell secreting signals that act on the surface receptors of the same cell type. So I just see it as it would be me talking to myself and performing what I said to myself. So you are sending out ligands that the receptors on your own body is accepting,
Yeah, but
right?
obviously it would also be on the same cell, but it could also be of same cells of the same type, right? So
Yeah,
it could be like clones of that cell.
right. And a good example of this is progesterone. I don't know if you guys know that hormone. We're gonna get into this hormone a lot because it does a lot. Appears to act in an autocrine manner in breast cancer, and in fact, autocrine signaling is very much a big deal in cancer because it sustains cell growth. And I don't know if you guys know, but cancer is basically abnormal cell growth that keeps growing and growing and spreading to other places. And a lot of times it's with the aid of autocrine signaling. Okay. So now we can move on to paracrine signaling. I hope that everybody understand that. That was easy, right? Right? We're good? I'm assuming
Yeah.
silence. Okay, great. All right, so paracrine signaling, no. Um, so simply diffusible proteins created by one cell is transported to interact with nearby cells over a short distance. So in this whole line, two things that I want you guys to focus on is diffusible proteins and over a short distance. So it's diffusible proteins that are so the two cells are talking to each other using diffusible proteins over a short distance. So this transduction pathway includes a signal molecule that we know is a ligand and a receptor. That's where the ligand binds to. And the receptor comprises of an extracellular domain, a transmembrane domain, and a cytoplasm domain. Simple. Okay. So diffusible protein and over a short distance. All right, next slide.
Oh, Tamara, can I add something here?
Yeah, no problem.
So I like to think of this as, your diffusible proteins. So think about diffusible proteins, they're being secreted. So it can't be over a long distance because then they'll get lost in transmission. They wouldn't go to the cell that they're supposed to go to. So they have to be nearby for diffusible proteins to work. So therefore it's a short distance. Just an easier way
Yeah,
to
that's
think of
good.
it.
That's good. Okay. So we're still on paracrine. It's more of like I'm giving you now an outline of how it kind of goes. It's really simple. Don't be intimidated by all these arrows, but taking it slow. Firstly, the ligand binds to the receptor, as you know, right? When that happens, the receptor experiences a conformational change within itself, right? And then the cytoplasmic domain is switched on. Remember, I said that there were three domains. You have the extracellular, the transmembrane, and the cytoplasmic domain. So the cytoplasmic domain would be the one that's inside the cell, and that would be switched on once the receptor has a conformational change. Okay, so after this domain is switched on, it causes a cascade of internal reactions. A bunch of things are going to go about inside the cell, and that ultimately activates a transcription factor. So, Ariane was mentioning transcription factors. So, I guess you guys can have an understanding, but if you don't, a transcription factor in the simplest form is thinking of something that controls the rate of transcription. Yeah, simple. And transcription in itself is changing DNA into RNA. So all of that can go on, and at the end, you can activate a transcription factor. And transcription factors itself can either inhibit or activate gene expression. Can look to the right, this cute little diagram again, where you can see the sending cell is releasing a lot of Okay. So to the right, I hope you guys can see my cursor, that the sending cell, this red cell is releasing orange circles. That would be the ligand or the signaling molecule to this green cell, which is the target cell. And you can see that the distance is short, nearby, and then the orange circles, which are the ligands, aka the signaling molecules. These are the diffusible proteins that I was talking about. So short distance diffusible proteins. All right, so a very major example of paracrine signaling is the synaptic signaling of the neurons, and I'm sure that you guys remember this, but just to go through it, you have the presynaptic membrane, that's the one before, and you have the postsynaptic membrane, right? So in the pre, there is the neurotransmitters. See them as the diffusible proteins or the ligands or the signaling molecules. They're in their vesicles and they're being released in the synapse right here. And then the receptors on the postsynaptic are going to accept these signaling molecules or the neurotransmitters, right? And it's paracrine because you're having the diffusible proteins, which are the neurotransmitters, and you can see the distance between the two cells, it's short. So if you want to remember, you can use the synapse as a good example for paracrine signaling. Are we good? Guys, let me know, if I'm going too fast, let me
Yes,
know.
we're good.
Okay, great. All right, so moving on from paracrine to juxtacrine signaling. People tend to confuse the two, but there's a clear distinction that we're just gonna break down. So unlike paracrine, the juxtacrine signaling does not include diffusible factors. So just remember that paracrine is the diffusible factors one. Juxtacrine does not. And it involves a protein from one cell interacting with the receptor of a nearby cell, or ligands that are present in the extracellular matrix that were produced from one cell interacting with the receptor of a nearby cell. So that's just cell signaling in essence, but juxtacrine takes it up a notch because there's no diffusible factors. So a great representation of this is actually cells transferring signals to each other via gap junctions. And gap junctions are like these channels that connect two cells, right? And the small molecules and ions can like pass through them. So they're not being diffused like in paracrine, where you see they're released into the extracellular matrix in a sense where they have to find their way to the target cell. It's more of like there's channels there, and that's where the proteins or the signaling molecules are transferring from one cell to the next. So that's the major difference, and also it includes direct cell contact. So you can see that the cell is literally right beside each other. Yeah, that would be juxtacrine. So gap junctions, juxtacrine. Paracrine would be like synapses, synapses. Yeah. All right.
Hello.
Yeah.
Yeah. Don't bite me for this for asking, but could you like slow it down a little bit?
Yeah, no problem.
All right. Thank
Do you
you.
want me to go back to juxtacrine?
Yeah. That. Yeah. That would be great. Thank you.
Yeah, okay. Aaron, can you go back over to slide 25? Aaron, is the slide frozen? Is everything okay?
Hi, Tamira.
Hey,
Yeah.
yeah, it's like first my Wi-Fi temporarily dropped.
Oh, okay.
What slide did you want to go back to? I couldn't even hear.
Oh, 25 to the one before.
Okay. Right, there we go. Sorry, I just couldn't hear the question.
No problem, but I'm not seeing it. Okay, great, great, great. All right, so hopefully it comes up for everybody now. So just going back to juxtacrine signaling. So as I was saying, that alpha people mix up juxtacrine with paracrine signaling. Guys, hold up for a second. Nathaniel, you had your hand up.
Yes, I wanted to ask if there was a difference in the range between juxtacrine and paracrine signaling due to like diffusible and non-diffusible factors.
Range as in how far the cells would have been between each other?
Yeah.
Well, yeah, because in juxtacrine, it typically is like a direct cell to cell contact. So they're typically narrow, so like right beside each other, like neighbors. And paracrine is more of a shorter distance with the understanding that they're diffusing, so they're moving along a distance, some form of distance.
Okay, thanks.
Yeah, you're welcome. Okay, so everything looks like it's back to normal. So just reiterating on juxtacrine signaling. So as I said, a lot of people do tend to mix up the two. Oh, yeah. So in paracrine, there are diffusible factors, but in juxtacrine, they aren't. And it involves a protein from one cell interacting with the receptor of a nearby cell, right? So they're right beside each other, cool. The protein, which will be in the signaling molecule, the ligand, whatever you want to call it, that's what's going to be sent from the two cells. Or ligands that are present in the extracellular matrix that were produced from one cell interacting with the receptor of a nearby cell. So this is like a general understanding of just cell signaling in itself, but with juxtacrine, think more on the lines of direct cell contact and non-diffusible factors. Those are like the two major things that should like pop out in your head. And as I said, a great example is with gap junctions, where there is actually a channel, and that's where the ions and the small molecules are passing through between each of the cells. Is that better? From John, I think John Pierre.
Yeah, that's good. Thank you so
Okay.
much.
All right. So some important things to note that weren't necessarily like in a themed where I could put down under a heading, but these are important to understand cell signaling a little bit better. So first of all, that in the extracellular matrix, there are a lot of molecules produced by cells residing there. And a good example of this is collagen. So cells in the extracellular matrix can produce molecules like collagen that are suspended in the extracellular matrix. Yeah. So these molecules can facilitate stability and migration. And if these said molecules are linked to cells by receptors, they can be called integrins. We're gonna see integrins pop up a lot more, so it's like important to just say, yeah, let me think of integrins right now, because further down in a lot of our other sessions, integrins is gonna be a big deal. This allows transmitting signals from the extracellular matrix to the cytoskeleton, right? So there's a linkage with some of the molecules are in the extracellular matrix, the receptors of cells, and because of that, they can transmit signals from the extracellular matrix to the cytoskeleton, which is inside of the cytoplasm of the cell. So also, migration with the usage of actin is possible because of these said molecules, and the integrins that we just discussed are important in influencing gene expression, right? Last thing to note that's really important is that signal transductions, they are not perfect, and a lot of our organ systems are not perfect, and oftentimes there's a lot of redundancies. So oftentimes they're gonna create stuff that are unnecessary, and literally if it wasn't there, it would not make a difference. But because of these redundancies, if there is like a mutation or if something happened, so a mutation or if there's a loss, it can be compensated for, or it won't result in like an abnormal development or death. So with all of these redundancies and all these unnecessary things, because there's a lot of them, if, for instance, one of them just cut out or it changed to something else, it won't affect the whole structure of um, the processing working of your body. So that's like an important thing to note. Okay, we can move forward. We're almost done, guys. All right, so in paracrine signaling, there are paracrine signaling factors that's important to note also. So basically, the diffusible proteins that we're talking about in paracrine signaling are called paracrine signaling factors. And a major example is ligand. And they're also known as growth and differentiating factors. So this is a lot of names. They have diffusible protein, they have paracrine signaling factors, and an example of that is ligand. And you have the GDS, They're all basically saying the same thing. So don't confuse yourself. They're all the same thing. It's just a lot of words for it. I don't know why. But yeah, diffusible proteins, paracrine signaling factors, ligand, GDFs. All right. So we're going to take on the form of GDFs for the paracrine signaling factors, the growth and differentiation factors. There are four major family groups of these GDFs, and namely it's fibroblast growth factor, you have WNT, and you have hedgehog, and you have transforming growth factor beta. So those are really the major ones to note. And for each of these families, they have specific receptors. So as we said, these GDFs are basically signaling molecules or paracrine signaling factors or diffusible proteins, whatever you want to call them. And all of them have specific receptors because signaling molecules bind to receptors. Got it? We're good?
Got it.
Yeah.
Okay. Okay. All right, so we can move on to slide 28. All right, so we're gonna describe the different paracrine signaling factors a little bit, or the GDFs. So the first one we're gonna go into is the fibroblast growth factor, FGF. There's not much things to know, just like a little intro, so you can have a feel of what it is, but it's not really anything high yield that you need to study. So they're named after the fibroblasts they created in the culture. Up to today, there's 24 FGF genes that are known, and the FGF proteins created from these genes target tyrosine receptor kinases known as the fiber growth factor receptors. So that's not really anything important, just like our intro, as I said. But they are important in angiogenesis, and I don't know if you guys ever come across this word, but basically it means creating vessels. Angio is for blood vessels and genesis is creation. So angiogenesis, creating of blood vessels. And they're also used in mesoderm differentiation. We're gonna get into what mesoderm is later on, but you have mesoderm, ectoderm, and endoderm, right? But FGF is specifically important in mesoderm differentiation. And that's it. So we can move on to slide 29. Okay, so the next paracrine signaling factor is hedgehog proteins, and this one's a little bit more important, so we're gonna spend a little bit more time on it. It's named after the bristles seen on the leg of Drosophila, and basically Drosophila is a scientific name for fly. So literally the bristles on the leg of a fly, that's how they gave it the name hedgehog protein. And there's three types of hedgehog proteins: sonic, Indian, and desert. But we're gonna specifically zone in with a microscope on sonic hedgehog proteins. So we're gonna start with why is it even important? Why are we magnifying it? So sonic hedgehog protein, or SHH, is a ligand, as we know, because it's a paracrine signaling factor, aka a GDF, aka a signaling molecule, also known as a ligand. So all of those names are saying the same thing. So ligands bind to receptors, right? So the SHH binds to the receptor known as patched, right? And it's on the cell membrane of embryonic cells. So SHH binds to patch. Patch receptor is responsible in inhibiting embryonic cell differentiation, right? And it also inhibits receptor-like protein smoothing. So patched, that receptor alone that's chilling on a cell membrane, once it's there, it inhibits embryonic cell differentiation. So it causes the cells not to divide. And also it inhibits the receptor-like protein called smoothing. Okay? Oh, and here's a little diagram showing the three different hedgehog proteins. You have a desert in green, Indian in red, and then sonic in blue. That's it. All right. So keeping the fact in mind that patched is inhibiting embryonic cell differentiation, and it also inhibits smoothing. So when
Wait, I'm
SHA,
sorry. I'm
yeah,
sorry. Could you go back to the previous slide?
yeah, no problem.
Right there where it says week 3, that means week 3 of embryonic development. So these signaling proteins wouldn't partake until week 3 of development.
Right.
Okay.
Yeah, so throughout the whole embryology course, there's different signaling molecules that come in at different times, and Sonic Hedgehog, we're gonna see further down the weeks that it does play a part in. But that's a good observation. That was Tristan, yeah. Yeah, that was a good observation, Tristan, yeah. So yeah, um when Sonic Hedgehog protein binds to the receptor known as patch, it causes the inhibition of patched. So can you imagine if it inhibits patch, what would happen? So since it inhibits patch, the inhibition of smoothing is now removed, and this allows the activation of genes that induces embryonic cells to differentiate. So because the sonic hedgehog binds to patch, you can now have an inhibition on patch. And remember that patch inhibited embryonic cells to differentiate, and it also inhibited the receptor-like cell called smoothing. So now that it's inhibited, we can have cell differentiation and we can have smoothing being able to do whatever it wants to do. So if you can look to the left and the diagram right beside the writings, you could see like the notochord cells releasing different hedgehog proteins, but specifically the blue ones would have been sonic hedgehog, right? So it's binding to this purple cell and on the receptor that's patched. So the SHH is binding to patch, right? And remember that patch inhibits cell differentiation and it inhibits smoothing the receptor. So when SHH binds the patch now, this activates genes for differentiation. So now we can have cells differentiating, and now we can have smoothing doing whatever it wants. Okay. Everybody gets that? That's fine? No, Ariane, go back to the slide. You guys are okay, right? You guys can say something, let me know if I'm going too fast. All right, I'll take it that everybody understands. Okay. All right. So that's one function of Sonic Hedgehog. The next function now, which is also very important to magnify, is that SHH protein diffuses with the embryo, and this forms a concentration gradient, allowing the embryonic cell to understand where it is in 3D space. And this is important as with this information, the embryonic cell can differentiate accordingly. So what all this is basically saying is that SHH forms concentration gradients, and because of this, the cell can find out where it is in 3D space. So if you can look on this diagram on the right of the writing, there is the notochord. Think of the notochord as like a spine or like a backbone to something, and you could see the little circles coming off. Those are the different types of the hedgehog proteins. We want to use the blue ones as sonic, the red as Indian, and the green as desert. So what's happening is they're being released, and with their release, they're in different concentrations. You can see that closer to the notochord, there is more of them, and as you go further away, you're seeing less of the sonic hedgehog proteins. Not the sonic hedgehog proteins, the hedgehog proteins in general. So, for instance, if a cell was to come near the notochord, like right here where that red part of the notochord is, it would it would be there and it would say, oh, I'm feeling all of these hedgehog proteins around me. There's a lot of them, so I know that I'm very close to the notochord. And with that information, they can divide into what they need to be. So they're very close to the spine, so they're gonna be like, okay, I'm going to form ribs now. Just like if they were further away where there's less hedgehog proteins, they would be like, oh, I'm not really feeling much of these, so I know where I am. Maybe I should be a leg. So that's how important SHH is, and hedgehog proteins in general. So they help the cells, the embryonic cells, to know where they are in 3D space. Because remember, these cells don't have eyes, so they have to use other things to figure out where they are and how to correctly form themselves. Are we good on that?
Tamara, can I just interject one more time? So you have the notochord, and Tamara keeps talking about the notochord. And throughout our other embryo classes, you'll see notochord quite a few, and you can think of this as your spinal cord. Because remember, fetuses or the developing embryo wouldn't have a spine. It would be this cord that first forms, which is kind of like what forms the spinal cord. And there's lots of divisions that take place within those weeks. But we want you to note that the notochord would be mentioned in other lectures, and that it is what will form the spine in the developing embryo.
Yeah, so notochord will be, I think it will be mentioned to the point where probably exhausted of hearing it, but as of right now, it's not a major thing. But Ariane is right. See it as the spine of the fetus and developing into that backbone that we know that we all have. So you can move on to the next slide. Everybody's okay, right? You guys are very silent. You can tell me.
Sorry, excuse me. Can you hear me?
Yeah, I can hear
Well,
you.
I was just asking if you could just go over this three-dimensional thing one more time. Just one more
Sure.
time.
Let me see if I can draw something on it. Um, okay. Okay, so Chelsea, imagine that I placed a cell here. Please draw. Okay, so imagine there's a cell right there, right? Okay. Um, the notochord itself is releasing these hedgehog proteins, right? And as I said, the blue would have represented the sonic, the red would have signified for the Indian, and then the green would have been the desert, because there's three types of hedgehog proteins, right? And while the notochord is releasing it, it's at different concentrations. So closer to the notochord, you can see that there's more of these circles here, and then further away, it becomes less and less. So that cell that I placed right there, close to the notochord, it's in that space and it's chilling and it's vibing, but now it needs to know where am I so I know what to do with myself. So how it does this is that it will perceive the outer environment, and it will it will it would feel like basically, okay, there's a bunch of Indian and desert and sonic around me. And we can even get more specific, like how much Indian is around me, how much sonic, how much hedgehog. Regardless, it's perceiving how much of the proteins is around the cell, and because of that, it knows that, okay, I'm very close to the notochord. So it has an understanding of where it is in 3D space. Because these cells do not have eyes, they basically use the concentration gradient of the hedgehog proteins to know where they are. And that's what I was basically saying. So if it was further away, it would have known like, oh, I'm further away. Shirley, you have a question?
Yes, I was asking if the notochord just produces hedgehog proteins or if it produces fibroblast growth factors also.
No, it doesn't produce fibroblast growth factors, but the notochord is known to do so many other things, and so many other diffusible things are going to come from it. So yes, you could just say that hedgehog is just one of them.
Okay, thank you.
Yeah, you're welcome. Okay. Chelsea, do you understand what I was saying? Are you good?
Yeah, man. Yeah, man. I understand. Thank you.
So all this is happening while the cell is a zygote, or is this like before?
This is more on the lines of after fertilization, and like you could say that the fetus or the growing thing is becoming something. So it's after fertilization when it's trying to form its arms and its legs and everything. So it's the embryonic cell.
Okay.
Yeah.
Hi, could you repeat what SMO or SMO is?
Oh, it's a receptor-like cell, and it's called smoothen, and SMO for shortening. That's basically what it is.
Okay, thanks.
Yeah. Yeah, it's not anything major. So it's just a receptor-like cell, and it's called smoothen, SMO. All right, so we can switch to the next slide. Um, just to note, the releasing of SHH is due to the works of the transmembrane protein called dispatched. Because you know that the ligands don't stay on the receptors forever. So specifically for the sonic hedgehog, for it to be released, it uses a transmembrane protein called dispatched. Okay. For the functions of Sonic Hedgehog, I told you guys two important, but it does so many things. It's insane. So left right axis formation, that's very important. We're gonna go into embryo a lot more. We're gonna see like there's cases where certain things that are on the left become on the right, and what's that called, and just the whole beautifulness of that, and how important it is to know difference between left and right as a forming embryo. It also helps with the midline, the cerebellum, and vasculature. So those are just things to note and have in the back of your head. Okay. So these two are not that important. I just wanted you guys to have like a simple understanding of at least something about these paracrine signaling factors. So for the WNT, thus far there's 15 genes discovered, and it's useful in limb patterning and midbrain development. Okay, nothing hard. That's it. As for As for the transforming growth factor beta, right? It's used in cell migration, so the cells moving up and down, and cell apoptosis. And I don't know if you guys know what this word means, but basically, well, that line is horrible. But basically, apoptosis is cell suicide or the cell off in itself. So that's what it is. So TGFB, transforming growth factor beta, cell migration, apoptosis.
Oh, and the formal definition for apoptosis is programmed cell death.
Right. Yeah. Yeah. Oh, I don't even know why I didn't say that. Program cell death. You're gonna hear that a lot. All right, guys. This is literally the last slide. Hold on. Yeah, this is literally the last slide, and it's just two other pathways that I think you guys should note. The notch pathway is a little bit more of significance, but regardless, I'm gonna start off with the planar cell polarity pathway. So starting here, the planar cell polarity, known as the convergent extension pathway. There is not much to know about this. It's actually a very hard concept to wrap your head around. Um, so I just gave you the basic baseline, and you know, we just move forward. So basically, the planar cell polarity pathway deals with the polarity axis that organizes cells in the plane of the tissue. So what all this is basically saying is that there's a polarity axis that is established, and it's used to organize cells um, to form into tissues in a way. So that's basically what it is. There's not much to say about it. So don't stress yourself. Don't think hard about it. Yeah, that's it for the planar cell polarity. Moving on to the notch pathway now, this is very important. We're going to delve a lot more into it in other themes and topics, but just a brief overview of it. It involves a transmembrane ligand of delta serrate lag2, which is a DSL family, and it allows the binding with notch transmembrane receptors. So as we know in cell signaling, ligand binding to a receptor, same vibe, same concept. Only thing, this ligand in the notch pathway is transmembrane, so it's gonna go through the membrane, the plasma membrane of the sending cell, right? And it also has a transmembrane receptor that's on the target cell. Okay, so when the ligand binds to the receptor, this binding causes conformational change, as we know it would. And in the notch protein, it's going to cause conformational change in the notch protein and a cascade of other actions. And these actions cause the DNA binding protein to remove its inhibition activities and allow transcription of notch genes. So again, we're seeing the ligand is binding to the receptor, and because of that, there is going to be some conformational change, and then in the end, we're gonna have transcription, whether it's gonna be inhibited or activated. But in the notch, it allows transcription, and it allows transcription specifically of notch genes. We're gonna get into that soon, but yeah, that's the overview. So don't stress it. Ligand, um, receptor, same concept, same vibe. Why is notch signaling important? It is used in cell proliferation, apoptosis, which we learned was programmed cell death, don't forget, and P and T cell differentiation in the immune system. In my little diagram on the right, yeah, you can see the DSL ligand. Remember I said that it was transmembrane? So you can see that it's going through the sending cell right here. Yeah. So the DSL ligand is going through the sending cell, right? And then on the other side now you have the transmembrane receptors. This is a target cell. Guys, don't mind my lines. This is really slow for me. But this is a target cell. And that's where the transmembrane receptors are on. And they'll connect and one bag of excitement will go on and allowing transcription of notch genes. And that's literally all you need to know right now for notch pathway. And that's literally all that you need to know right now for cell signaling. Are you guys good? You guys have any questions?
Hi. Are you
Hi,
hearing me?
Janelle. Yeah, I can hear you.
Okay, so the notch pathway and the so the notch pathway is just used for pancreas signaling, or both the pancreas and the juxtacrine signaling?
It's not necessarily labeled under those, so that's why I kind of put on the other pathways.
Oh, okay.
Yeah, so it's just like other, they literally have specific names for themselves, and these are it. So you have the planar and the notch. So don't put them under paracrine. Okay. Okay, good.
Thank you.
No problem. All right, so that's it. You guys are good. Guys, tell me if we're not good. No, it's fine. Okay. All right. I'm assuming that your silence is that you're good.
Okay, so guys, this is the end of our recording. We're really happy that you all sticked around, and we just included a few tips and tricks right before the slide that we wanted to let you guys know so you could navigate Embryo a little bit better.
Yeah.
And we really hoped you enjoyed the class. What did you guys think of the class? Could give us feedback.